Welcome back, everyone. Please join us in welcoming Dr. R.K. Choudhary, Scientist E, Agharkar Research Institute, Pune, who is a renowned expert in plant taxonomy and molecular systematics. With over 15 years of dedicated research, Dr. Choudhary has delved deep into the intricate world of plant diversity, exploring the rich biodiversity hotspots of India, the Eastern Ghats, the Western Ghats and the Andaman Islands. Having earned his PhD from the prestigious Botanical Survey of India and Rajiv Gandhi University, Itanagar, with a focus on the floristic diversity of the Upper Siang district, Arunachal Pradesh, Dr. Choudhary's expertise is further enriched by his postdoctoral research experiences in South Korea and Vietnam. Today, we are privileged to have him share his insights and knowledge with us. Now I would like to request Dr. Choudhary to please present his talk.
Thank you, Shabna. Such a great privilege to be here to my alma mater, Botanical Survey of India. To me, it's like coming back home. And when I stand here, I remember I was getting very nostalgic thinking about my JRF days. I was the Flora of India scholar here in the Botanical Survey of India. I had a very strong connection with this. So once again, thanks to the organizer for giving me the chance and to discuss whatever we have done in the past few years. So today's topic is little from like morning session we had a lot of discussion on morphology. Now we'll be moving towards molecules. And post-lunch talks are always tricky. So it's a bit risky also for me, but I will make sure that we're still awake. And where is the pointer? Yeah, thank you. To start with, I would like to a little bit introduce, I don't know how many of you know ARI, Agharkar Research Institute. Any of you know this place? ARI, one of you, right? So just would like to introduce it as an autonomous institute under Department of Science and Technology, Agharkar Research Institute, and probably, I'm not sure, but probably the only institute in India named after a plant taxonomist. Right? Am I right, sir? Is there any other institute named after Birbal Science Institute there, but he was not a plant taxonomist, right? Is there any other institute you know? I also want to learn it. No other institute in India which is named after a plant taxonomist, right? So I feel very privileged being here. And here in ARI we worked under 6 different themes, starting from biodiversity and paleobiology groups, big group. And then we have a bioprospecting unit. Another group work on bioenergy, microbiology. Then we have a nanobioscience group as well, and developmental biology group where we work on 3 model organisms, zebrafish, Drosophila, Hydra. And then we have a big genetics and plant breeding group also, where we have another unit where we work on 3 different crops. We have several ICR funded projects. We work on grapes, wheat and soybean. And here you can see Professor S.P. Agharkar, who actually finished his PhD with Angler, German, the Angler and Plant System classification. So he has done PhD with him. And when we went to Germany, that time World War was broken, and he was put under jail. And in all these tough circumstances, he finished his PhD, returned back to India, joined Kolkata University, I think he's in presidency there. And then after retirement, he came back to Pune, put all of his money to establish this institute, Agharkar Research Institute, which was earlier not named as Agharkar Institute. In his honor only, later on the name was changed, and it was called as the Agharkar Research Institute. So this is the history. I hope you will have some interest and welcome to visit there as well. And there we have a biodiversity and paleobiology group where we work on plants as well as diatoms. And we not only do documentation work, rather we also try to understand evolution, explore it for human welfare, try to have some conservation activities, and also try to communicate the knowledge which we learn, which we do through various activities. So my flow of talk, I divided it into 4 parts, as I was given a very tricky title, whether molecular taxonomy, which I have been doing for the last 10 years, whether it's a boon or bane, though it is written ban there. I don't know, it's printing mistake, but whether it's a boon or ban. So first half, we would like to focus how it is a boon to the taxonomy, and when can it become a bane. And then also another thing Das sir has added, their implications on species concept. So that was very tricky. Also gave it a thought how I should present it to BSI. But before I start, I would like you to go through a paper by Hasse et al., which was published in 2009, which says nature itself neither needs categories nor has any knowledge of them. Categories are artificial and always delimited by an individual or collective convention. Getting it? When we delimited something, we make classification system, it is done by either one individual or a convention of people like APG. It's a convention of people. Or Bentham and Hooker system of classification. Two people are involved, I think, Bentham and Hooker, Engel, Prandtl, Taxonomy. So it's decided by one individual or a convention of people. Humans make and define botanical terms and we use them to facilitate communication. Right? It's for our convenience, the classification system. Although all too often we take them to be real. There is the problem. Many of the times we think what system I'm proposing, it is the real system. But there the problem lies. It is a dynamic thing. With our improving knowledge, it changes. It's a dynamic system. What Bentham and Hooker proposed, that was also not accepted, appreciated by many of the people. What Taxonomy proposed was also not appreciated by many of them. So because we started thinking it is real, and they can be as much as an impediment. Because when we think it is real, that becomes the impediment to our understanding as anything else. So just keep this in mind because this is the theme of my talk. And another famous quote I would like to have mentioned here by none other than Wallace. You all are aware of Wallace, right? No species or genus occurs in two very distant localities without also being found in intermediate places. Getting my point? If it is in Tamil Nadu, if it is in Maharashtra, or it is in Kerala and Maharashtra, possibility is there, it will be somewhere there in Karnataka also. Okay, so this was pointed out by Wallace long back in 1855. And still, this is making a lot of sense. Agree with me? Yes. So before I start, I would like to ask you one question. Who were our ancestors? Can you raise your hand and tell me? Who were our ancestors? Just on a funny side. Nobody knows? You were right. Right? Monkeys, in layman terms, we call monkeys, right? You're right. Why it is important to know? Our ancestors. Why it is so important? It is important because studying our ancestors from a scientific perspective allows us to trace the evolution of the species, identify the inherited traits, and gain insights into the adaptation of a species to different environment. Agree with me? That's why overall, this ultimately contributes to our understanding of evolution and the forces that shaped our biological and behavioral characteristics. Agree? If you disagree, just raise your hand. You're free to ask. I have 1.5 hours or more than that, so we can have a lot of discussion. Now, I have another question. Who were their ancestors? Before I go ahead, I would like to have one, I think, just knowing about your background. So is there anyone who is into biotechnology or into this? How many of you are doing this traditional taxonomy, morphology-based taxonomy? Could you raise your hand? Okay. And how many of you having any genetics background? Okay. So I just want to know so that I can mold my talk in that way. So I'm going to ask another question. You know that our ancestors were monkeys or whatever. Who were their ancestors? You know this plant very well, right? Neem. Who were their ancestors? Okay. Prokaryotes. So that way. Okay. Who were their ancestors? Apple? Any idea? Who were their ancestors? Rose? Any idea? No idea? Okay. Who were their ancestors? Lemon? Now, looking at this, we should be thankful to this DNA, which tells us who were our ancestors. Monkeys were our ancestors, we know because of the similarity of the DNA which we have. Same way, if we are making use of DNA, we can very easily know about the ancestors. So this is how we come to know that apple, sweet apple, is very close to rose, and that bitter neem is more close to citrus. Right? Rutaceae. Right? Rosaceae. Getting my point? So, I'm going to talk a little bit about DNA-based classification system or phylogeny, with which we know that angiosperms are more close to gymnosperm, and how they have evolved, at which rate. Using morphology also we can do it. Right? But this is the question of precision. You get accurate knowledge, information. This was the first question. Now, this molecular systematics. First thing, any of you, I would like to ask you, can you just open this bottle, okay? I'm not able to open it. What would be your approach of opening this bottle if it is very tight? I'm not able to open this bottle, or there's a screw, I'm not able to open. What you will do? With my hand, I'm not able to open the screw. Yes, you will bring a screwdriver, right? Whether I am doing something different, I am still opening that screw, trying to open the screw, I am still trying to open. So molecular systematic, you have to first think, it is not a different science. It is also about doing taxonomy. Altogether, our aim is to understand the same thing, right? So once we are not able to solve any problem using morphological approach, we make use of anatomical data. Could it be cytological data, embryological data, or molecular data, to understand the same thing in a better way. Right? Agree? So it's not a different science. Many people have that apprehension. So you are very new to BSI, you should not go ahead with the same apprehension. It's a tool. First thing, you make it very clear. And here, what we make use of, the genomes on plants. Where you will get it? In plants? At 3 different places. They are either in nucleus, or in chloroplast, or in mitochondria. Okay? Getting it? Agree? Animals, they don't have mitochondria and chloroplast, so we make use of the nuclear data. And we already know about the discovery of the new species. We have been describing the new species for quite long, but suddenly there is a change. There's a very fast it has become. You see, there is a report. We are discovering and naming new species at a much greater rate than any other time during taxonomy's 250 years of history, maybe more. And also around 2,000 new species of plants and fungi are found each year. This is a report from RBGQ. And we should be thankful to the new tools and techniques that we have, which are helping us in describing and understanding the species in much better way. Now, this is a paper by Howrah et al. Whatever work you're doing in BSI, you think, right? You understand that you are contributing to the knowledge of biodiversity, right? How many of you think that whatever I'm doing, I'm contributing to the knowledge on biodiversity? You think that is true? Are you contributing a little bit to biodiversity? Yes or no? Raise your hand. Yes or no? All of you think like that. So, but before you think so, the first part, as I said, is older slides. Many of you have heard this, including Abhishek. So, but the second half, totally new, exclusive to all of you. So those who have heard all these things can simply revise, brush up. So, if you're thinking that I've contributed to the knowledge on biodiversity, read this paper by Howrah et al. published in 2015, which talks about 7 different shortfalls. First shortfall is lineage shortfall, which tells unless you describe each and every species on earth, there is a shortfall called lineage shortfall. Until you know about the geographical distribution of the species, there is a shortfall called Valesian shortfall. Until you know about the populations of the species, there is a shortfall called Prestonian shortfall. Until you know about the evolution of the species, there is a shortfall called Darwinian shortfall. Until you know about the functional traits and ecological function, there is a shortfall in your study called Roncarian shortfall. Until you know about the abiotic tolerances, there is a shortfall called Hutchinsonian shortfall. And if you don't know about the ecological interaction, still there is a shortfall called Eltonian shortfall. So molecular taxonomy or molecular tools help you to overcome all these shortfalls. At least all these first four which I mentioned, they are directly related. All of them are actually related to each other, but they will help you in understanding or overcoming all these shortfalls in a much better way. And there is also many of the times the enigma of convergent evolution. We know the presence of wing in bird and bat. Are they very closely related? Bird and bat? No. But both of them have wings. Taxonomically they are following the same group. Same way? No, right? And here same way I have shown you one euphorbia which is almost looking like a cactus and another cactus. They are looking similar. Are they the same ancestor? They have same ancestor? No. How we can find it out? DNA will help you in finding it out in a much easier way. Right? Then there is a problem of cryptic species diversity. What are these cryptic species? When you have morphologically, they have a look-alike population, but when you check it at genetic level, their DNA, it shows a lot of differences. Such species are called cryptic species. Okay. So, is it not our duty to know about the cryptic species or the genetic diversity present in those species? Why I am saying this? Because if the diversity, the genetic diversity is less, they are more prone to extinction. That genetic data we have already proved. There are so many papers available. That if the genetic diversity is less, that species is going to go. But if the diversity, genetic diversity is high, they are going to exist here. That's why it's also our duty, being in BSI, to know about the genetic diversity. How much diversity within the population, within the species exists, so that we can predict whether there is a threat to those species or not. Okay? So, this is an example of a particular species. It was considered as complex. Recently they described 11 species from one complex. From China, of course, in 2020. And they use all tools and advanced techniques. They describe 11 new species based on the genetic diversity analysis. Sorry, the lunch effect is more on me, it seems. Okay. So what we do here, we make use of several genetic markers. And these could be 2 or 3 different types: morphological, biochemical, or molecular. In molecular also we make use of 2 different kind of markers: hybridization based or PCR based. In PCR we make use of PCR. I think most of you are aware of it, called RPD, FLP, SSR. And you have site specific markers like ITAs, RDCL and all. And then you have hybridization based marker like RFLP. And they all just to resolve the complexes and to understand the diversity. This is, I would call, the superhero of genetic markers, ITS we call. This is not a gene, it's an interspecific region. And then it's found in nucleus. Small region, just 600 to 800 bases long. But they're very much conserved to the species. So it will be different. Suppose we have 2 mango species, one is from here, one is from Andaman, they will show differences. Mangifera andamanica, Mangifera indica, whatever. So they're very much conserved to the species. This is one of the very small region from nucleus. because sequencing the whole genome is very expensive, time consuming. So we are making use of this information. You see, sorry, this probably is not, this region 5.8S, 18S and 28S. These regions are very much well conserved to the species when you look very detailed of it. Same way, in chloroplast also you have several regions which are very much conserved for the species. Like RBCL, MADK, they are found here, small region. Instead of sequencing the whole genome, you can simply sequence this small region at a much lower cost, 300, 400 rupees, and you can find the identity of the species just by comparing the sequences. Same thing in mitochondria also, you have several regions. So basically what we do, what we do here, we isolate DNA from the plant, amplify those particular regions like ITS, RBCL, what we're using PCR, amplify it, send it to company, they're going to send you sequences, and the sequence comes, check it, and you can compare, you can construct phylogeny, you can try to identify the species at a very accurate level. This is the basic structure of molecular taxonomic studies. And then this is how you do it. You get your DNA isolated. In this DNA you have DNA from nucleus, you have DNA from mitochondria, you have DNA from chloroplast. Add your primer, ITS suppose you want to identify. Add this, put in master mix, do PCR, send it to company. Very simple, not very complicated. Send it to company, company is going to send you sequences like this, check it, how good it is, and you have to curate it. That's very carefully, you have to check it, and you can construct your phylogeny. Okay, these are the very basic steps. Those steps, some small steps are involved. Just the basic framework I have showed you here. So the word cladistics actually comes from a Greek word called klados, which was given which means branch. And this concept actually long back proposed by an entomologist, Mr. Willy Hennig. 1955, he proposed this in 1950 in his book, saying that it is, and the same book was published in English also, 1966, and he has recognized this cladistics, and he has said it is one of the approaches of biological classification in which organisms are grouped together based on whether or not they have one or more shared unique characteristics. Okay, we are not going to discuss all these things today, but you have some basic understanding what was the meaning of cladistics. But later on it was appreciated by people from different streams also. Like in plant taxonomy also we started adopting this idea, and then we started making use of this cladistics or systematic studies. So first I'm not going to, that's another lecture, I'm not deviating. How it is a boon. Later on we'll discuss bane as well. How it is a boon. The first example I would like to give is APG system of classification. Many of you might be disagreeing with the classification proposed here, but you try to understand it's a very long process through which it has undergone and it has evolved. And also many of you think it is also not of practical applicability, but the whole world is now following the system of classification. There must be some logic. So at this stage you understand this is the present system which we are following. It has gone through a rigorous process, and then we have come up with this phylogenetic tree called APG4, where we almost know about the ancestry of 99.9% of angiosperms called mesangiospermy. So out of that, total we know about the phylogenetic position of 2,95,781 species. What we don't know still, because the reason I'm not going to study, called ANA group. Still we are unsure of their phylogenetic position, and this includes members from umbrels, nymphials, austrobilels. Altogether there are 183 species, but we have a very clear understanding of their ancestry now. Okay? Try to make sure, just understand, it is not a tool for identification of the species. It's not like Botanical system classification, which actually tells you how to identify the species. Here we are trying to find out the ancestry and how they are related to each other. That was the purpose of APG, not like helping you in identification. Okay? First thing which has like practical application, and first thing which was useful in understanding classification. Now I have another question for you. What is this plant? Okay, what is this? Anybody who don't know this? All of us we know, right? What is this plant? Very good. Now you tell me, what is this plant? No answer, right? Why we are not able to answer this? Yeah. So don't you think that is a limitation of traditional taxonomy? Right? Don't you think? Do you agree with me? Can you identify this? Yes or no? Loudly. No, we can't do. Tell me. You can't do it. And worse it becomes when people come like this. Whether it's a gold milch or papaya seed, you don't know. And it goes worse when people come to you asking to identify this. Can you do this? I can do this. Don't you think it's a boon? At least up to genus level I can identify this. So don't you think it's a boon? Yes or no? And you will be surprised to know that many of the rare plant species from our country are being sent to another country in this form. We don't know it's a Terminalia or Olphia serpentina. There's putting level of Terminalia, and from our country Olphia serpentina. There is no technique actually which can tell it up to species level. You do HPLC, HPTLC, LCMS, it will still tell you about the whole profiling. Yes, this particular chemical is present, but species level identification would be very difficult. But with DNA, our database is so rich nowadays, at least definitely you can reach up to genus level. Isn't it a boon? Yes or no? Isn't that a limitation of traditional taxonomy? Can you identify this using well-known Hooker system of classification? No. Shall I move ahead? So now with the help of DNA, we are able to develop DNA barcodes. And when I talk about DNA barcodes, don't think that still simply amplifying one region and telling this is the barcode is correct. It's again a long process of establishing barcode, DNA barcode. It's not like simply amplify ITS and say, okay, I'm able to identify, so this is a unique barcode for this. Okay, that we can talk someday, the other day. Right now I'll focus on this. Enjoying this? Okay. So it helps you in DNA barcoding. And with DNA barcode, what we can do? We can properly identify the species, we can properly classify them. Also it helps in conservation. I know one friend, I'll just tell you a story, true story. I'll not reveal the name of the friend. He got a DBT project, almost 50-60 lakhs rupees. Project was approved, funded, very well executed. So many research papers came out. Right? Incidentally, we went to collect the same plant on he had that DBT project. And when we checked the plant, identity was wrong. So all the papers published from his lab on the plant, it was wrong. Because the identity was not confirmed properly. And that was a very taxonomically challenging group. So in such cases, unless you prove correct identity of the plant, you get a certificate from somewhere saying that your whole project can have a totally different results. Right? So that way. And also, in US several cases have taken place where you are conserving a wrong species because you don't know the identity of the species. They spent millions of dollars on conserving species. Later on, they came to know they were actually conserving a different species. So it can help you in reducing the wastage of money as well. Okay? Agree? Okay. I'll take your consent then and move ahead, right? And barcoding also has gone ahead. Earlier it was up to some loci-based approach we used to follow. Now we have gone up to genome level. Okay, even in plastome based approach we follow, which we call super barcode. Like you can consider the whole genome and then they develop a barcode. I'll tell you the story of our work which we have done. This plant you might be aware, called Periocolon. You might have seen this. And if you go to Maharashtra, there's a very famous UNESCO heritage site called Khas Plateau. You'll see it's a very beautiful site. If you have not visited, you should consider visiting. And you will see like whole bed of these kind of white color flowers here. And these are the Eriocolon. And this area is very rich, biodiversity very rich. And you see all Eriocolon how they are here. And there you can find 4 different species of Eriocolon. And one of them is strictly endemic to this particular area. But on a herbarium sheet, if you want to identify them, you have to dissect the head. Small head it has, tiny one. And these 3, taken from 3 different herbarium sheets. Can you identify this like this? Which species it could be? Can you say? Does it look different to you? All looking same, right? Yes or no? Yes. So if you want to, and the worst part, say many of the species have medicinal property. One of the species is very much effective as a neuroprotective agent. But how to identify which one has that particular chemical? You have to have proper identity of it. And in India you have almost 100 species of it, restricted to northeast India and Western Ghats, mostly. Also many have properties against sinusitis, eye diseases, and dental treatments, headache. So if you wish to identify this, first you have to dissect the head. You have to check for the character of involucral bracts. Floral bracts, male flower, which are very tiny, female flowers, but that's not enough. You cannot confirm the species identity yet. You have to check for the seeds. You see how big it is. And light microscopy is also not helpful. Even SEM, when you do scanning electron microscopy, you have to look for the appendages characters. So, and it shows lot of diversity in appendages. So unless you check each and every character, you cannot confirm the species identity. So that's why this particular plant group is known. And also within species it shows variation, and different species show very less variation. That's why it is also known as taxonomist nightmare. So Hooker, when he was writing for a British India, he has written, they are the most difficult of classification, presenting no good sectional characters. We thought, why not make use of these tools and techniques what we have presently? So we started working on it. We wanted to collect it from all over India and whatever the occurrence region. It has a distribution in Meghalaya as well. We collected a lot of sampling we did through Western Ghats and also in Meghalaya and Northeast region. We collected more than 550 excisions of these. In India we have almost 100 species tentatively, and we were able to isolate DNA from everything, and we published this finding where not only we synonymized 7 different species which were actually pseudo species, I would say. All of them we could not collect out of, but our sampling was almost 70%. So these species we synonymized, and also we reinstated some species based on our finding. Not only this, we now understand how they are phylogenetically related. So this all findings were published here. You can find this paper here. If you don't have, I will be happy to share. And then it shows little congruence with the previously presented taxonomic classification. Still a lot has to be done, but this is one of the way molecular data helped us in understanding this plant group. Another group we worked on is called Capparis. Here we have moved ahead. In the world they are known by 140 taxa, and in India we have 35 species. So there, they don't only have taxonomic issues, but also we wanted to understand the phylogeny. We also wanted to understand its biogeography, and we also wanted to do DNA-based barcoding. Why barcoding was so important here? Because, sorry, this Sanjay ji. and this understanding the phytogeography. We wanted to do this. So with the help of all our collection, we could not only produce this phylogenetic tree which suggested us to reclassify. And also, if you know, there were several species complexes were there which we were able to resolve, and all the findings we have published here, how DNA data helped us in resolving these all species complexes. And another thing, why I was saying DNA barcoding was important, because one of the species called Capparis spinosa is the most important ingredient of this famous drug called LIP52, Capparis spinosa. But there are so many other species looking like this, so unless we identify it up to correct level, or most of the companies, they'll come with us a powder form. In that case, you cannot identify this spinosa or zeylanica. So if we establish DNA barcode for this, within a day you can identify, you can plant very precisely. Right? Wasn't it a boon for this group? Yes or no? Then we also worked on several species complexes which are already published here. I've just shown you this paper, how we have established DNA barcode. If you want to learn more about barcode establishing, you can go through this paper, which are there, and also the findings were published in this. How it has helped us in not only understanding the phylogeny, but the whole biogeographic study. The details are discussed here. And here our sampling was from almost throughout the world. We had certain collaborators in South Africa, we had collaborators in Vietnam, so we collected samples from all across the world. Another paper, as I said, it becomes a boon when we have to deal with the cryptic species diversity. Why it is important to understand, we have already discussed, because we have to know about the genetic diversity, which are under threat or which are going to be here, to understand that how much diversity within the group is there. This example I have given you, where very nicely they have done this study and they have proved how they could be different. And nowadays also many people are working on it, trying to understand the cryptic species diversity. And nowadays we have next generation sequencing in hand, where at a much cheaper cost you can do whole genome analysis, whole genome sequencing. Earlier, whole chloroplast genome sequencing used to cost you around more than 1.5 lakh rupees. Nowadays you can do in 25,000 rupees, whole chloroplast genome sequencing. So we have made use of these tools and techniques also, not only to identify, but also to establish DNA barcodes. And you see several kind of studies, because it has become much cheaper. Here, the Sarella, a very famous fellow of grasses, he has published a species based on 250 plastomes. 250 whole chloroplast genome study they have done. And after APG was proposed based on small region like ITS or BCL, it was proposed. But these fellow, they have done whole chloroplast genome of most of the representatives of angiosperms, and then they have produced a copy of APG classification. They wanted to check whether there is valid classification or not. They have also come up with almost similar pattern, saying whatever was proposed in APG is correct. They have made use of 80 genes from 4,792 chloroplast genomes. Okay, this very exhaustive data, and belonging to 4,660 species, and they have produced this tree, and this was also similar to APG4. Not only this, there is a recent study published by Howrah from China again. There they have done this using whole nuclear genome analysis they have done. And still the tree is having the same shape. APG, if you know this anagrades there, the maglodides, magnets, whatever there, still the same. Earlier people had a lot of apprehension. Okay, the APG is based on some small regions, so it's going to change. But when they checked at the chloroplast genome level, it was same. They checked it whole nuclear genome level, it is still same. So isn't it a boon? We got some structure, we got to know something. Do you agree with me, or you have any apprehensions in your mind, something running there? You can ask me here. Okay, I want you to be awake. Some people started sleeping, I can see. Okay? Getting my point? Getting my point or no? Yes, say yes or no. Okay, shall I ask you something? Who was the person who proposed the term cladistics? Sleepy? Yeah, that group is more active, seems. Okay. It was very Hynek, who was an entomologist basically. And not only that, when it becomes a boon, when you want to identify, this was a very famous study produced for coffee. You know the coffee, why we feel, when we are sleepy, we wish to have coffee, right? Why we wish to have it? Because that contains caffeine. And that triggers your mind again, you become so active. These fellows that worked on coffee, they wanted to understand how this coffee has evolved in all these species. They were surprised to find out almost 50% of coffee species, they don't have caffeine. Only 2 species which we use, they have highest amount of caffeine, which are like commercially utilized. Here you can see the red color line. It shows the highest presence of caffeine. And the same way, blue line indicates no caffeine. So how it has evolved, they simply map the data on a phylogenetic tree to understand the evolution of caffeine. So this kind of work you can also do. Nowadays so many free softwares are available. You simply have to explore a little more and you can do similar analysis, publish very high impact papers. Isn't it a boon for scholars? Yes. And this one, another paper published in Impact Factor 8.45. This by very famous group called Donoghue. He's a polygonasy guy, very famous molecular taxonomist. And they picked up very small problem. There were 3 different species and there are a lot of fight among taxonomists saying no, they all belong to one species. Some group know that there are 3 species, some saying no, 2 species. These fellows, they simply use some new techniques to prove the point that actually there are 3. And they could publish, their analysis was rigorous, that's why they could publish in a systematic biology. Earlier it had 10 impact factor, now 8.5. But very nice study they have done and they were able to prove like how the serration how this data, they have used rat data and they could confirm the identity of the species or these complexes. And most importantly, when it is a boon to us because we are able to publish high impact papers. That is another requirement, right, for our promotion. Agree or not? Yes. So these all papers, they use simple taxonomic problem, but hi-fi some little advanced tools and technique. It's not very hi-fi that you cannot do this. In your lab you can do using very freely available softwares. Okay. But everything has pros and cons, right? Being a molecular taxonomy, I should not only praise this. I should tell you about the negative points as well. So I have created this section for you people when it becomes a bane. Do you think like it can also be bane? What is your opinion? I want to make you awake. I just want to interact a little bit. I would like to ask you a bit. So your opinion about molecular taxonomy from this side who are more sleepy, can you just tell me something? Why do you feel molecular taxonomy should not be appreciated? Yes. Costly. Yes, that's a valid point. Of course. You? Costly, of course, but some of the times you have to think of because I'm not telling for everything you do molecular. When you have some complexes, you are not able to resolve using traditional technology, then you use, and if you are proposing the same thing to some funding agency, they will also be happy to give you money. Okay? If tomorrow you are saying, Mao sir, with this problem we are not able to resolve, none of the previous taxonomies were able to do this, I want to do it, I'm very certain, definitely he will give you more money. Okay? You can see Abhishek is sitting here. He got whole molecular lab because of this survey. Right? So anything else you feel like why it should not be appreciated? So I'm going to tell you something. When it becomes a bane, you are very much hesitant. Am I very like I have something here. You don't want to talk. You should speak. Okay? Yeah, please. You can sit and talk, no problem. Yeah.
Yes. We can easily identify and keep the record of
Yeah.
particular taxonomy, if we
Apply, yes.
there will be a lot of confusion among the people.
Yes.
Like some species will go to other family,
Mhm.
that will go to other family.
Okay.
If we draw the that thing.
Mhm.
So according to me, I think there will be
The practical applicability is less, okay? Because in field you can simply recognize it's a gamopetly, polypetly, easy. That's more easy. But as in the beginning only I have stated, it is not a tool for identification. APG system is not a tool, it tells you about how they are related. It's a phylogenetic history. It's not a tool, it's a tool for not for identification. For identification, you are learning in a much better way here how to identify a plant, how they are grouped, how they are evolutionarily related, that it tells you. Okay? But still, there are some glitches which I am going to discuss with you. Yes, please. Yes. 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 Mm-hmm. Yeah, I got your point. Yes. Yeah. I got your point. You're very correct. Okay. Yes. Yeah. Good journal. They will do some basic molecular turn. Yes, sir. Yeah. So of course, when you are doing it, do it perfectly. Otherwise, don't do it just for the namesake. Okay. So today, without wasting, I don't know how much time I have. I think I have enough time, right? So when it can become a pain, when it makes you more uncomfortable, just see the first point. The first thing, as he was mentioning, if your sampling is poor, you have been assigned to work on Ranunculus genus. Okay? You know that it is in Kashmir, in some interior locality. You simply go up to Delhi or somewhere, Himachal and all. Then, no, I have funding constraint, I don't want to go to Kashmir, come back without collecting that species. So that never works in this phylogeny. Your sampling is very, very, very important. Sample as much as possible and complete range you will have to complete. Suppose if it has 40 species, or making it for all 40 together, then it is a true phylogeny. Otherwise, still you have some missing link, it may be a misleading phylogeny, which many people in India they have done earlier. I don't want to name them, but there are several studies published here, which actually now in today's perspective you see they are kind of rubbish. They are not revealing the true phylogeny. So first thing, the most fundamental issue is the sampling, taxon sampling. See this example. This is they are dealing with some 4 species. They collected all species from India and they are telling the phylogeny is like this. A3 is more related to A4. Okay, they are done, they have put in thesis, it is published. But some other fellow, he collected A3, another allied species from Southeast Asia somewhere. And this A4 also he collected some which are allied species. Now you see, earlier studies it says that A3 is more close to A4. But when your sampling has improved, A3 is telling that A3 and A4 they have different ancestry. So the story is totally different. So if your sampling is erroneous, if it is not proper, you are going to produce something like this. And you are going to interpret in a very different way, which is not correct. Right? Got my point? Yes. See, the same story I have highlighted here. So second point, if you do not select a suitable marker, as I earlier showed you, ideas, RBCL, MATK, how are you going to make sure that this is a suitable marker for me? Somebody told you use RBCL, I take RBCL, you are not getting any resolution. So, there is always several kind of markers available. I don't know if you know about this coding and non-coding regions. So there are 2 kind of different markers available in the market. One are we call neutral markers or non-coding regions, which actually they don't code for any protein And here markers under selection. Markers like tRNA, tERNH, PSBA, they are useful in a very peculiar cases when it is a very like recently evolving clades are there if you are dealing with. So before selecting your markers, you have to review the literature very carefully. Then only you decide which marker you have to use. So many of the time we are confused. Sometimes we make use of just nuclear marker. Sometimes we take, okay, I should use plastid marker only or not, because earlier people they have used only plastid, so I will use the same. If they have published something bad, you will be simply following it because your review of literature is not proper. Or I should use the combination of markers. These kind of things, if you are not sure, are simply producing a phylogenetic tree, sending it to sequencing company, they send you some sequence and you are tick, your phylogenetic tree is ready, send it for publication. Next day, rejection comes. Don't do that. And which combination is suitable? So you have to do a lot of review of literature and also consult experts. This is the first thing. Also, if you do not select the desired number of markers, sometimes sir, ITS is not getting amplified, so I am ignoring it. Sir, RBCL is not getting, I am ignoring. Don't do that. Include as many as markers possible. And see the cases, phylogeny based on a single marker is often not a good estimate of the species phylogeny. Sometimes we just simply want to produce a tree based on single marker. Okay, it is ITS based, this is a good marker, you write anything you want, but that is not correct. See, there are two things. One is called gene tree. Am I too heavy for you people now? Too heavy? You want a tea break? Okay. So one is called gene tree, another is called species tree. You should be aware of this. When you are just producing one tree, the seed, there are 4 gene tree I have shown you here. Like this could be for ITS, RBCL, MATK and some PSB. 4 different markers. And this is called species tree. When you put them all together, this is going to give you a better resolution. So gene tree may not truly reflect the correct phylogeny. Always keep that in mind. So if your phylogenetic study is based on just single marker, that not be the true phylogenetic study. So that's why always you go for the as many as markers possible. And third one, if you do not know, I don't know how many of you earlier you have any exposure to molecular tools and techniques. Can you raise your hand? I will have an idea, just I'm telling something like that. Do you have any exposure earlier to this molecular tools and techniques? 1, 2, 3, 4, 5. Last workshop you were there, right? So you cannot say no. So we should understand, when you receive sequence from the company, what we do, we use it as such. We don't bother to check the sequences, what they have sent it. The process is called curation. Once you receive the sequence, you have to check how correct it is. Don't simply rely on the sequencing agency. And whatever they send, produce your phylogenetic tree based on that. There are several studies who have simply done it. Don't do that. So, when you receive it, the sequence, you have to do alignment. Put all sequence together, all species, and perform a test called sequence aligning. After aligning only you can produce a phylogenetic tree. So, you have to know about the gaps and missing real data, the missing data. There's always an option. Shall I treat missing data as gap? We don't know anything. You'll say, go over default option, produce your tree. Don't do that. If you are doing this, you are going to again produce a wrong tree. So it needs a kind of expertise. With a lot of experience and analysis, you will learn all these things, how to treat these gaps, or whether I have to treat this as a missing data. So see, I have shown you example. When you receive sequence, it will appear with you for species 1, 2, 3, 4 here. So here you can find one gap. So where I have to shift this, delete this gap or keep, everything has to be known perfectly. Looking at the chromatogram which the company also provides. We never bother to check that chromatogram. Simply whatever they send, we use it and produce our tree. Don't do that again. It's a sin. A real gap is one where one or more sequences have an indel. Indel means insertion or deletion. And when you have the missing data is always coded as N or what. Whenever you see in your sequences this N or what symbol, you have to very, very carefully and then check the chromatogram, then only you finalize. We always generate a consensus sequence and that only we have to use. It's not like simply click, click, click. What we generally do, produce a tree, sir, this I've got, it appears to be a new species. Many people have done that. That's wrong. That's a sin. And the worst part, if you are treating this N as a gap, there are two ways of generating tree, ML algorithm called maximum likelihood and maximum parsimony. So MP has a different way of interpreting the things. The algorithm will take that as missing data. Another one will take as a gap. So both of them, the algorithm will result in different trees. Now you are confused what to do, what is the correct relationship. But you don't realize what mistake you have done in curating your sequence or alignment. Right? How many of you have performed all these while dealing with the molecular data? Very few we do this curation properly. That has to be done accurately, otherwise it will interpret with the tree badly. Then, if you are unsure which tree building method to use, during lunch break I was asked a question. I am trying to generate a UPGMA tree and I am using different bootstraps, and every time it is giving me different bootstrap. I don't know whether you are aware of these things, but that is another. I found out there is some the problem with the basic understanding which tree I have to use while generating this. There are two ways of generating trees. One is distance based, another called character based. Distance based is to understand the evolutionary distance, the distance of the sequences. Another one is character based, where we make use of sequence information, whether there is insertion, mutations. But here how much distance is there, that we calculate like UPGMA and NJ, and then we generate the tree. Here ML, Mr. Base, they are all character based methods. So once you have data set, you are unaware whether I have to use UPGMA, I have to use NJ, because the basic concept is missing. Many people, they simply produce UPGMA and they think this is a correct tree. But we have to understand what is the difference between them. Then only you produce it. Okay? Yes, please. Yeah, distance-based method. Okay. There basically two, you must have heard about UPGMA, unweighted pair group method while using algorithm, what is that? Arithmetic mean. And NJ means neighbor joining tree. There, it is there to understand the genetic distance between two sequences. Suppose you have sequence from one family called rose, another from apple. You want to understand the genetic distance, like based on that, it has a formula which calculates how much indels are there, how many mutations are there, and it will calculate and give you a mathematical value. Distance between these two organisms in 0.23. And based on that value, it generates the tree. But in character-based method, you can find out whether this is a mutation or insertion or deletion. Is there a point mutation there? You will understand with that. So they all methods, they have a different algorithm of constructing the tree. We don't understand that, we simply click, 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 go. Got it? Yeah. Okay. Anybody has any confusion, they can raise hand and ask. Yes, please say. Yeah, sure. Yeah.
Usually, I'm trying to borrow a particular from I understand. Basically, when we do the regression analysis, we have something called aspect statistics.
Yeah.
So if we have the suppose 100% aspect is all points are falling on the line of best fit.
Correct.
Do we have something like that here that will tell us that a score that whether is sufficient or not? And second, can we also link this with the idea of can we use a bootstrap value as a proxy for R square statistic equivalent? Because my sense is that if there is insufficiency anywhere, that should reflect in poorer bootstrap values.
Yeah.
So if you are saying like you
It should be at least 70% or more. Don't go on statistical analysis since previous. If you are doing that, may end up messing up the things. Okay, don't do. And the second part is whether bootstrap is a proxy for that. No, it is not. Bootstrap, if you can do between two different closely allied sequences of 4, 5 also, it simply bootstrap means what? If it is 100 bootstrap, it means it performed the same analysis 100 times, and out of all 100, it got 100 times the same result. It does not mean, it will not indicate whether your sampling is poorer or strong. It will simply tell you these two sequences, they are alike or not, or they are simply alike to each other or not. Okay, so don't use bootstrap as a means for that sampling. Okay. Yes.
So, in that, the best choice will
It depends on your data set and what actually you want to do. It's not like for each analysis the same method will work. Okay. If you are interested in understanding the genetic distances between two organisms, then I would suggest you go for NJ method, where you don't need an outgroup. But where if you want to understand the evolutionary pathway, in that case I would recommend you to use ML or Mr. Bayesian inference. Okay, that will detect the evolutionary pathway using your outgroup. Okay, yes. Anything else? Yeah, sleepy? Yes. Okay. So shall I move ahead or want to break for tea? Okay, na? Okay. You want me to finish fast? Okay. Okay. Because more complicated things are ahead. If you want a break, I can give you. But it will help you in understanding the developing molecular taxonomy is not as simple which you think, like simply take sequence and generate a tree. That's true molecular taxonomy. No, it is not. Yes, please.
This is a phylogenetic tree. This will create only a phylogenetic tree.
Yes.
And taxonomy is the composition of three things: identification, nomenclature, and classification.
Yes.
It will not help in identification. We need to identify.
Really? What then I was talking about DNA barcoding.
Yes, in identification, if I don't know one single species, if I want to make a tree and I need 100 species, so I have to first identify that those 100 species are in my
Yes,
genome.
correct. You have to have DNA of all those 100, then all you can tell. But yes.
If we first identify those by genome, that we nomenclature is not needed basically. Should it be called a molecular taxonomy or molecular classification?
That's a molecular systematics we call. Systematic studies mean what? Systematic taxonomy. It has been used in different way by different people. First thing you should understand the purpose. I want to open the screw. I'm not able to do it with the hand, so I'm using a screwdriver. Molecular systematic is a screwdriver to you, which helps you in opening the screw. Right? So nomenclature problem, when you have the problem of correct identity of the species, right? Then you go with the nomenclature, okay, this was looking like this, type was missing, this and that. I'm coming to that also, how it will be helpful. So indirectly or directly they are related with correct identification of the species, right? Then you see with the type specimen, many of the type specimen identification itself wrong. Many of the cases we have, right? So in that case, what you are going to do, if you are able to isolate DNA from the type species, can't it help you in resolving the case? Yes. So it's a screwdriver. I'm not telling you it's a totally different science. Okay. Many people have this mindset only. That's the problem. Okay. So today I'm going to talk about this screwdriver. Somebody from cytochemistry or cytology, they will come. That's another screwdriver. Somebody will tell you embryological data. That's another screwdriver. Okay. Yes, any more? Yeah. You can sit and talk, no problem. Yeah.
information is different. They are saying that it is.
Yeah. If there is a conflict, right? So, okay, let me finish because more complicated things are there, right? So let me finish and then I will be able to answer this question. Okay? That's you are raising a valid point. Yeah. Okay? So shall I move ahead? Are you getting it? Okay. Whenever you need a tea break, just raise your hand and tell me I need a tea now, it's too much. Okay? So the correct tree, like which method to construct the tree is very important. Then accuracy of the phylogenetic reconstruction. You have reconstructed a phylogeny, but are you sure that phylogeny is correct? There's a quote, it says it is impossible to know the true phylogeny unless we have the ability to go back in time. I am saying that okay, we have evolved from monkeys. Whether I have the ability to go back in time and see whether we had a common ancestor? No. We cannot go back in time, right? That's why true phylogeny can be produced only when you have the ability to go back in time. What we are doing based on nucleotide information, what we get, and based on certain algorithm in the empirical evidences, what we see how much mutation has taken place between these two individuals. And one more thing, another thing you keep in mind that in molecular taxonomy or systematics, we have the concept of OTU, no concept of variety, forma or subspecies. Each and every individual is taken as operational taxonomic unit. Okay, so don't talk in that term. Always take every individual is an OTU. So, accuracy of the phylogeny construction. Sorry. Yeah. This, you have the sequence data with you. Now your alignment is ready. You want to simulate the data. Put in a computer, ask your computer, I want a maximum likelihood tree. Okay? And computer will execute that, simulate the data, and then it will apply various models of evolution. It's very important for us, a person who is doing phylogenetic analysis, very important, very important I would say, to know about the models of evolution before going to construct a phylogenetic tree. What are these models of evolution? Any idea? Because you must know about it. If you're planning to do this, just I've shown you here, I will show you. It's a model of evolution. What we see, if you have just two different sequences, one is telling ATCG in Mangifera indica, in Mangifera andamanica it is telling AGCT. So it means there is a substitution of a base. Some base point mutation is there. Now just look at this case. You have one sequence here, ATCCGGTT. And with this we understand the species is 50 million years ago. This is old. This was we just identify like this using some data, fossil data. It is old. And then now in the same species you have found a species, same group, which has one mutation T. Instead of G, T has gone. So now what I presume, for G to become T, it takes 50 million years. Can you predict like this? Can I predict like this? Yes? Sure? Yes or no? Loud? No or yes? Why? Here I have a species having G, and after 50 million years I have T. So I am saying it takes 50 million years to become this G to be converted to T. Why you are saying no? Sure. How you can make sure of it? You were there in previous workshop, seems. So you are thinking right. So in 25 million years ago, this G got converted to C. And then after 25 million years, your assumption now changed. If I am telling it takes G to T to convert it to 50 million years, am I right? No. Because it could have happened that it converted to C 25 million years ago, and the C has again converted to T. So now what I am presuming, that it takes 25 million years total time to become C to T. There are two kinds of mutation, right? Transitions, transversion, right? Pyrimidine to pyrimidine, purine to purine, like that. We already know. And they have different way of changing. Like it takes a lot of time. There's already set formula. I'm going to talk about this. So now this changes. So how to resolve this problem? How you can predict like this in phylogeny? Can you do this? Is it correct way? We must have some mechanism to resolve this issue, right? So that's why this nucleotide substitution or understanding of models of evolution is very important. Okay, see another case here. I have A, T, C, C, G, G, T, and I know it was 50 million years ago, right? And now what I see, it is A, T, C, C, G, G, T, T now. There is no change in 50 million years in time of time. So am I correct? So they are all conserved. I'm saying during all these 50 million years ago, they were conserved. They remained the same. Am I right? That's why. Huh? Yes or no? Huh? Loud. Okay. So see another case. 25 million after, this got changed to C, and then C again got changed to G. So my assumption again is wrong. Right? So it could be or could not be. To resolve this issue, several models of evolution have been proposed by various scientists, which we call as the models of evolution. So just like when you go through construct this, it will ask you which parameter you want. Whether you want JC, Jukes Cantor, you want Kimura 2 parameter, you want Kimura 3 parameters, or what. There are more than 60 models of evolution proposed. And then when you are analyzing it, you have to have the selection of a proper model of evolution, then only you can produce a good phylogenetic tree. So how to resolve this? And this model, suppose Jukes and Cantor were proposed in 1969 long back, and they have assumed all substitutions are equally likely. It means C can be converted to T, T can be converted to G, G can be converted to C, anyway. They're equally likely. It was not appreciated. Then somebody from Kimura, Japan, they also proposed there will be one transition rate and one transversion rate. If it is purine to purine, that rate would be same. If it is purine to pyrimidine, the rate would be different. Right, same. Then this was also not appreciated. These fellows, they proposed another thing, Kimura 3 parameter. And now what we use, we don't use this. Just in a simple manner I have shown you these models, but there are so many models of evolution have been proposed. So how to select this? Answer please loud. How to do this? How to overcome this problem? Because we don't have the understanding, basic understanding this, that's why we simply produce anything, any kind of tree. That is, and that's why you think, okay, that lemon will go with banana, banana will go with mango somewhere in your tree you're producing, and there is a conflict between traditional taxonomists and molecular taxonomists. Because those people have not done it properly. That's why there is a problem. Got it? Yes, sir.
Alright, is on fine.
Yeah.
Your tree is constructed on the assumption that the evolution has taken only by mutation.
Yes, sir.
There are a number of genetic problems.
Correct.
What happened to that?
Sir, I'm coming to it. Sir, can I?
It is a one-sided story.
Yeah. Can I just go ahead and then complete this story?
Yes, you complete the story.
Yes.
What I want to say is that mutation is one.
Sure. Yeah, you're right, sir.
We know it once they named it mutation.
Yeah.
By the rapid mechanism.
Correct.
Where within 8 years I find variation.
Yes. So, can I finish this, sir? Yes. I have tried to answer those things as well.
You are not simply spending much time on your mutations. You look at the other variations which will expose your barcoding. I have a basic question.
Yes.
Your barcoding.
Yeah.
In an outbreeding species, there is an explosive variation from generation to generation. It has been demonstrated that the F2 generation variability is much higher than the variability that exists.
Yes, I'm coming to that, sir. with humble request, would you please listen to me and then I'll go ahead. Yeah. Yes, I'm going ahead because the story, everything should be clear. Yes.
The audience will get an impression that evolution has taken place only by mutation, my phylogenetic thing is all.
No, because my story is not complete. Yeah, my story is not complete, sir. Just 60%. Just I'm coming to that. I'm discussing the boons or banes. Okay? So I still have to discuss the how implications of species concept. So this was the three. Understanding the models of evolution is very important. First thing. Now comes what would be the implications of species concept. Okay? So let's start with the fourth one. Species concept and species delimitation. One of the concept was typological species concept, right? Where it has been said that a group of individuals that differ from other groups by possessing constant diagnostic characters. And here it was based on collecting and describing a type specimen, right? You should have a type sheet or type specimen for that for a given species. But what problems were there with typological species concept? The polymorphism within the population was ignored, right? If you have a type but polymorphism, how much it shows, it is not reflect on the type sheet. Is it so? Just one sample, right? Another one, geographical variation among population. One sheet collected from Andaman, and it has been found in Sri Lanka also with some variation. Whether the type is not reflecting, can you describe it as a new species? There is a flaw. And the cryptic sister species. If there are some genetic variations, we cannot resolve that. And then convergence issue. We have discussed that, showing the case of presence of wings in bat and bird. Another concept called biological species concept by Dobzhansky and Meyer. Species are the largest and most inclusive reproductive community of sexual and cross-fertilizing individuals that share a common gene pool. Agreeing to that, Meyer also says species are a group of actually or potentially interbreeding natural populations that are reproductively isolated from other such groups. This also has several problems. It is not applicable to asexual species. If I want to describe a Hydra species based on this concept, can you do that? They reproduce by asexual means. Can you describe a Hydra species based on that? No. Reproductive isolation is often incomplete and it is difficult to verify whether they are reproductively isolated or not. Third concept, which was given by von Wellen, ecological species concept, it is a set of organisms that is adapted to a particular set of resources or niche in the environment, which explains differences in form and behavior between species and adaptation to resource availability. But this concept was again, it cannot be an exclusive diagnostic of the species. Just based on ecological variation, you cannot describe a new species. Degrees of ecological preference again is very important. Third concept, another concept was by Simpson, evolutionary species concept. Evolutionary species is a lineage evolving separately from others with its own unitary evolutionary rate and role and tendencies. And then this also had some apprehensions, I would say. It was initially developed to define fossil species only, and how you define independent roles and tendencies in that. And does not provide a mechanism to understand this. So there is for each and every concept there is a flaw. Another one is phylogenetic species concept. There is another flaw also. It says the smallest diagnosable monophyletic group of populations within which there is a parental pattern of ancestry and descent. Again with the problems, what characters to use? How do you deal with reciprocal monophyly at the population level? I'm going to discuss this also, what is reciprocal monophyly. And the gene trees versus species tree. If you are just producing a phylogeny based on a single gene and multiple genes, that is also conflict is there. So this concept is also not appreciated. Overall, just you have to remember 3 points. Morphological divergence, which we usually feel is not a proxy for speciation. Ecological divergence alone is not a proxy for speciation. High P distance is proportional distance when you are comparing two different sequences. These high P distance values are not a proxy for speciation. And then phylogenetic bifurcate. Each branch need not represent a different species. Now you will think you have been saying about these phylogenetic bifurcations, and then again you are saying it could not be the proxy for the speciation. Why so? But you remember one thing, and then this also tired seems. So, a lot of development and concept, This paper you should go through by VNs, which says there have been real progress made, and there's a lot of brainstorming in understanding the species concept. So he says there's been real progress made in thinking about species concept, which now makes some general agreement seems possible. As evidence of some of this progress, there seemed to be a general agreement among almost all participants that species are lineages. And Deaker's paper, which comes in 2007, the most recent species concept till now by Deaker, which says various species concepts such as BSC, PSC, ESC are related to diversity of events or sub-processes that occur during speciation, and all modern species definitions are variations of the lineage species concept or general concept of species. But altogether, you will see that all species concept, they are agreeing to a particular point. That under all concept, a species is a separately evolving metapopulation lineage. Metapopulation, what does it mean? It's a metapopulation lineage, separately evolving metapopulation lineage. Metapopulation means if a species from here in India, same it is distributed till Africa, that will is considered as a metapopulation. Okay, so that is they all are agreeing to a particular point that saying the species is a separately evolving metapopulation lineage. That's why sampling is more important here. Okay, now since I'm dealing with Boon, and before that I would like to discuss this also, the concept. Till now, the most recent species concept given by particular science is called Deaker. It's freely available online. You can read through it. Species concept and species delimitation. And it says the property acquired during lineage divergence it has considered, and these properties are not necessary and definitely not sufficient to delimit species. I'm going to show you the big picture of it, saying this. What they have proposed, how a species is evolving. Here, the paternal species was there. And you can see the species criterion. Criterion here is given, species 1, criterion 1, 2, 3, 4, 5. And this whole zone is called grey zone, where two different species are hybridizing and ultimately producing two doctor species. In between this grey zone, what is happening here? First instance, the criterion is there will be diagnosable differences. It could be genetic divergence or differences or morphological differences. Second one, complete cessation of inbreeding. The inbreeding will be ceased here in this stage, and then monophyly one species. So this is now evolving as a distinct species, and it is also dividing. So one species is given rising to this. But this grey zone is very important to understand what is happening here. And then when it is under different stage of evolutionary divergence, it will exhibit different level of allele sorting or lineage sorting in the nuclear and plastome trees. Because we know about the alleles, how the allelic variance, it does not necessarily reflect the phenotypic characters. So Deaker says geographic information is also crucial, because nearly all species exhibit geographical variation, and it is possible for large difference to exist between populations within the same old and geographically widespread species than between populations from different but recently separated species. And here another approach, which if you want to study your species evolution perfectly, But we know there's an approach has been introduced called the coalescent approach. Is it too heavy? Yeah. So coalescence is just opposite of divergence. Divergence is one species is evolving like this with time. Coalescence is something going back in time. And looking how this, suppose if there is going means studying that grey zone. Studying the grey zone, how this evolution with time and space, how it is. I'm not going to go in detail because it will be too lengthy for you. But try to understand this coalescence. And this was based on a model proposed by Wright and Fisher long back in 1930. It says, suppose this is a population of species, if with time it is evolving like this. Can you see blue color here? So some allelic differences have come in this population. This has increased here a little bit. Again it gets lost. This is a natural phenomena in nature. Allelic differences. Then again there is the emergence of one allelic difference here. In one population this also. So they randomly appear. Based on different factors, ecological factor and some other factors. And then see how this one allelic difference is moving ahead, leading to evolutionary of a distinct lineage. Getting my point here? And our effort is to understand and to go back in time. Believe me, that is possible now with a lot of analysis. So, to go back in time, you just see how we call it a reciprocal monophyly, how they are evolving. Go back in time and try to understand how the alleles, they are going ahead. And there is a lot of calculation which the software will do for you. This is called multi-species coalescent. And that is nowadays is kind of very hot topic of discussion to understand the species delimitation. So I'm just skipping these all probabilities, but if you see how this coalescence works. So you have 3 different species, you're going back in time with allelic variation study and trying to understand the ancestry. This is called multiple species coalescence. Okay, so I'm skipping this. With this you can do this, understand ABC. And this you see a species tree has 3 different species and the genealogy of single gene in context of the species tree. This color each dot represents the allele present in a single individual and row represents a discrete unit of time. This. Ultimately we go back and try to coalesce that and understand the variation. So Dickeridge also proposed this, how either they are reproductively isolated, so there is an interbreeding, like hybridization is taking place, and there is a 4 tests you will have to perform. If you really want to go back and try to understand, we call intergradation. And using those data, molecular data and analysis, you can understand how this hybridization and intergradation, how much it is taking. Okay, I'll finish in 10 minutes, I guess. Okay, so it's taking place. So analysis can go up to that level. And I will show you some examples also. So this is called intergradation. See, hybridization as species taken between 2 species and then intergradation. Intergradation means here some flow of the characters. And I'm talking about the grey zone, always keep in mind. Not a distinct species when it has been formed. So how this grey zone intergradation has taken can be mapped. And based on how the species are evolving, but always keep in mind that lot of population, genetic, like population sampling has to be there in that case. Exhaustive sampling is very important in this case. So we can try understanding that. And there are several softwares like that. General Mix Yule Coalescent using R package, you can study that. You can understand Bayesian Poisson tree process is there using online server. It's freely available, you can do that. Also using some software in Beast, you can do that. And it works on several statistical model which will, with the help of your nucleotide data, it will go back in time and try to analyze how much substitution and what are the factors. Everything you will have to put there as an input. And then you understand. And many times you have to include fossil data as well. And then you try to understand the evolution of species. And there's these approaches or the software which can be used to understand this. But this also has several limitations, and as you can see, there is a famous paper on Hemidactylus, this kind of gecko. This was published in for zoo taxa, and very famous, I think synthesizers know this. So they established a new species of gecko. It is just some personal communication with the authors. They established, based on this divergence, they established a species, saying there are a lot of complication in problem with were there in morphology based taxonomy, so they found divergence here. I'm telling you the negative side of it. They found some variation and they established a new species. But when the authors, they went ahead, they did a lot of sampling across the occurrence zone. Like this was the sampling they did when they were doing phytotaxa, zootaxa paper. But when they did real sampling throughout Western Ghats, they found that was not the real new species, which they have not published. Probably they might publish someday, but it actually defied that concept. So that way it could be a pain as well. That was my point. So you have to be very careful. So a new species is not always a new species if you have single divergence or similar. I'm just answering your question, sir. So this was the case. That could not be always a new species. That's why your proper sampling, proper analysis is very important. Before saying it's simply based on molecular data and proposing a new species, that's not always correct. So they did, it's based on personal communication. They've done this. And as I'm not going to talk the apprehensions and challenges, but also it is based on concept of monophyly. So I'm saying here, how many species do you see here? So this, you can treat this as a monophyletic group. You can also treat this as a monophyletic group. So which has to be considered, that's always a question. That's another negative point of phylogenetic approach. Then no interspecific variations can be seen many of the time, which we consider as between species there is some variation that cannot be seen several times. And then the effectiveness of species delimitation tool is determined by the data you supply. If you're just giving data for 70 species, you cannot understand phylogeny of 100 species, right? So amount of data is very important there. And another aspect, cannot account for reticulations between the tip nodes. How much gene transfers are taking place? We already know about the horizontal gene transfers. So how much it is, it cannot tell you about that. These are the limitations. And there is I have discussed the multi-species coalescent. There is a paper in PNS, already very reputed journal, and they have also argued that that is not the way to delimit species. So they have given the title multi-species coalescent delimits structure, not species. So they have argued and they have not accepted the way multi-coalescent approach. So this is another side you should also know. So being honest, I'm not favoring molecular taxonomy. Whatever has been done, whatever way it has been argued is not effective. Both the things I've proposed. The argument here, given if you can go through and read this paper, you will find their logic is also correct that way. So go through this, and then the question they have raised, our work raises question about the practice of genome-based species discovery, which cascading consequences in other fields, specifically all fields that rely on the macroevolutionary dynamics. They're saying that maybe if you follow this approach, instead of one species, you have 10 different species. That will inflate the number like anything. You might be aware that cobra, it was earlier used to consider one species. Now we know cobra, king cobra, is known as 6 different species. So that is, with some analysis, you are inflating the number. That was their argument. So if you do this much analysis, it's a genetic structure. Based on genetic structure, if you are trying to define one species, that is also not correct. That is their argument. Okay, which I have kept in front of you. Now it is up to you, how you take it or you don't take it. But if you take it, write in a paper with a lot of arguments, like what we consider, why we consider this as a new species. With all such analysis performed, you will be able to publish in a 10 impact factor journal, definitely. I can assure you. So what to be done now? You said you are just telling the problem, but not the solution. Second last slide. Okay. First thing, thorough sampling across the distribution area. Many of the times we simply don't do sampling, proper sampling throughout the area of occurrence. Then multi-gene phylogeny. Don't consider just one or two markers. Do as many as possible. Various multiple species coalescent, that model you have to consider and to test if they are sexually isolated, hybridizing or genetically interbreeding. These all parameters if you consider, and then you decide whether your species is a new species or not, then definitely you will be able to publish that in a very high impact journal. So okay, with this I am concluding my presentation with one message, that for the great majority of taxa, the alpha stage has not been passed. If you are doing flora work, revisionary work, particularly sitting in a country like India, Cambodia, Laos, Vietnam, which are biodiversity very rich, that is also appreciable. That's also an addition to science. And what I've talked about here, this alpha taxonomy and this, the new taxonomic approach, it's not necessary that all the times you have to go with that. So if you're doing this alpha taxonomy, that's also fine. But you have to be a careful, thorough and competent descriptive taxonomy can reveal fascinating details of the evolutionary history. What I am just pleading, that you have to keep yourself updated. You must be able to understand what is multiple species coalescent. You should be aware of it. And what does it mean? Whether I should perform this test or not. So Rao sir, in one of his papers, he has written, is it the time to become chemocyto-histo-eco-morphomolecular taxonomist? My answer is yes. We have to be smart and we have to be like this nowadays. It's very competitive. Many countries are moving much ahead of us. We have to know about the chemotaxonomy, we have to know about the cytology, we have to know about histology, everything. Then we decide whether my species is a new species or not. Blindly don't go. These frogs are talking. It's not whatever what is my morphology. We're not talking. It's like looks are not matter. Looks aren't everything. It's what is inside you that matters. Okay. With this, I would like to thank everyone, including Director BSI, Dr. S.S. Das, who gave me the chance to speak here and to discuss whatever I know. Now it is up to you, the way you want to take it. Okay, thank you so much.